Welcome back to the deep dive. Today, we are taking a stack of source material that, well, it sits squarely at the intersection of biology, computing, and, yeah, outright science fiction. And we are going to extract the startling truth. The technology for targeted systemic human evolution is kind of already here.
It really is.
We are wading into the world of nanites, those microscopic self-replicating machines. But we're focusing on the ones nature already perfected billions of years ago.
Viruses.
Exactly, viruses.
And that's the critical pivot point, really, for this entire deep dive. For most of human history, the virus has been the ultimate, unstoppable destroyer, just devastation.
Right, feared above almost anything else.
But the material we've synthesized for you today, it completely reframes that. It treats disease not as physical damage or infection in the old sense, as a software malfunction, a bug within the body's primary operating system. So our mission today is to go through the functional blueprint that shows how these biological destroyers can be, and frankly are, being converted into microscopic molecular couriers. Scribes, if you will, designed for healing, regeneration.
And eventually.
Yes, intentional human augmentation.
Okay, that changes everything. I mean, if disease is just a bug, then a cure is just a patch, like a software update for your body.
That's the core analogy.
The mission, then, is to extract the nuts and bolts of what the sources boldly call programmable evolution. And here is the initial sort of stunning premise. A virus, functionally speaking, it's nothing more than a highly efficient protein shell carrying a payload of instructions, DNA or RNA.
Just a delivery vehicle with a message inside.
So if we, the biological engineers, replace the malicious hijacking code that causes illness with constructive therapeutic instructions,
then that virus instantaneously transforms. It becomes a nanite of healing. And what's so compelling here, is that we are not talking about inventing some complex machine from scratch out of silicon or metal.
No.
We are simply repurposing the best, the most refined delivery system ever created. Nature did the hard work.
We're moving from fearing nature's programmer to, well, co-opting its expertise.
Exactly. Learning from the master, you could say.
Okay, let's delve deeper into that idea then. Viruses as natural nanites. The source material is pretty emphatic about this.
It is.
Viruses are not clumsy, random invaders. They are biological machines refined over billions of years specifically for genetic delivery and for reprogramming host cells.
They're specialists,
Yeah.
microscopic specialists in infiltration and, crucially, payload delivery. That's their whole game.
And this isn't just theoretical stuff waiting on some, future breakthrough.
Not at all. This is happening right now, just maybe at a smaller scale than the full vision. We are already using engineered viral vectors in gene therapy.
Right, for specific genetic disorders.
Exactly.
Yeah.
Delivering small corrective genes to patients with specific inherited conditions, that's established practice.
So the next step is just scaling up the complexity.
Pretty much. The goal is to direct these systems to carry out far more complex tasks. Think repairing multiple genetic mutations at once, or regenerating large volumes of damaged tissues.
Like after a major injury.
Or even, eventually, entirely restructuring failing organs. Imagine rebuilding a heart after catastrophic damage. The fundamental delivery architecture. It's already functional. We know how to get the package there.
And the power of the payload they carry, the message inside. That's why this is such a transformative idea, isn't it?
Absolutely.
We are not just talking about data in the traditional sense, like ones and zeros. We are talking about life's operating code itself. DNA.
And if we're going to discuss programming life, we have to talk about the storage medium.
DNA.
DNA, it's the code of life, obviously. But it is also, according to the sources, the densest information medium known to science, period.
The metric they use is humbling, like you said.
It really is. We are talking about, well, incomprehensible density. 1 single gram of DNA
Tiny amount.
can theoretically store over 200 petabytes of data.
Petabytes. I always have to remind myself how huge that is.
For perspective,
Yeah.
the source points out that this capacity exceeds all the digital photos and videos currently residing on planet Earth.
All of them in 1 gram.
All of them. Think about the entire accumulated visual history of the digital age, contained in a pinch of organic material. It's staggering.
I mean, I had to look up how big a petabyte was again just to try and grasp the scale.
Yeah.
That means DNA isn't just dense, it's also incredibly stable, right? Much better than silicon chips that degrade.
Far more stable over long periods, yes.
Yeah.
And crucially, we know this isn't just theoretical storage capacity. This isn't just math on a whiteboard.
Okay.
Scientists have already proven this. They've encoded arbitrary data, entire books, high-resolution images, even films.
Actual movies.
Actual moving pictures, encoded into synthetic DNA strands, and then they successfully read that information back using standard sequencing equipment.
So the read-write process is established.
It is. And the logic follows. If we can write a movie into DNA, we can certainly write a therapeutic program into DNA.
That capability, that's the pivot point that radically reframes disease, isn't it?
Completely. If we accept DNA as programmable, customizable software like, the biological equivalent of Java or Python,
Yeah.
then disease fundamentally becomes a software malfunction.
A glitch in the code.
Exactly. We shift the whole paradigm. We move from just treating symptoms, masking the problem,
To debugging the core code, fixing the error itself.
precisely. Instead of simply managing the effects of the runaway program, we attack the underlying code error.
Let's take cancer. We traditionally see it as just uncontrollable cell division, cells gone wild.
Right. But in this framework, the source material describes it as a corrupted loop.
Like in computer code.
Exactly like that. The cell forgets its instructions, it ignores the molecular stop signs, and it enters an infinite destructive replication loop. It's not that the cell wants to grow uncontrollably.
Its internal operating system is just glitching catastrophically.
That's the idea. And that logic, it applies across the board. Think about autoimmune disorders.
Where the body attacks itself.
Yeah, horrible conditions.
Right.
They're described not as, say, a failing organ or a simple mistake, but as the systems attacking themselves because their identifying code has drifted.
So the immune system gets confused?
The security parameters get corrupted. The body's defense mechanisms misidentify self as foe. The whole system needs an immediate security patch update.
Wow. So the solution becomes, well, almost elegant and direct.
It is. Viruses, reprogrammed as these nanites, they become the delivery mechanism for patch updates.
And they don't just offer temporary relief like a painkiller.
No, they bypass the downstream damage entirely and deliver the necessary new code to rewrite the underlying biological system. Restore the cell to its intended healthy programmed state.
So the doctor of the future, they might prescribe not a traditional pill from the pharmacy,
but a bespoke custom DNA patch, tailored to your specific genetic glitch.
encased inside a tailored nanite.
Delivered directly and only to the affected cells, minimizing side effects.
That is truly a foundational revolution in how we approach human health. It's profound.
Right, and to execute that level of precision, moving from broad chemical fixes, carpet bombing the body essentially, to this kind of surgical code delivery, the nanite needs a highly structured operating system. Can't just be dump and run.
Needs rules.
Exactly. And our source material details a potential blueprint for this. They call it the tri-mode architecture.
Dry mode.
This breaks down how these new viral nanites would actually function therapeutically. They act less like a simple chemical and much more like a sophisticated, targeted mini-computer programmed for dynamic control inside the cell.
So this is where we move beyond just simple delivery, just dropping off the package.
Right.
And we establish molecular mission control. 3 modes? What's the first one?
Well, the first mode, the one that probably generates the most immediate interest, especially for diseases like cancer, is the seek and destroy mode.
Sounds direct.
It is. This mode is specifically designed for eliminating rogue cells, primarily cancerous ones. The nanite is engineered to identify specific molecular markers.
Like little flags on the outside of the bad cells.
Exactly. Unique surface antigens only found on cancer cells, like a molecular license plate. And once that target is confirmed, this is key. It doesn't just poison the cell indiscriminately.
What does it do?
It delivers a very specific kill switch payload designed to trigger apoptosis.
Apoptosis. That's programmed cell death, right? The cell kind of dismantles itself cleanly.
Precisely. It's crucial. It's not a violent death that causes massive inflammation and damages nearby healthy tissue. It's a clean, quiet, controlled self-destruction. Much safer for the surrounding environment.
Okay, but I have to ask, how reliable is that targeting? Cancer cells can be sneaky. They might look a lot like healthy cells. How does the nanite tell the difference with enough certainty?
That's a critical question. And the precision is engineered into the system itself. It relies heavily on 2 components we touched on earlier. First, highly selective viral vectors choosing the right envelope that naturally prefers certain cell types.
Okay.
Second, and maybe more importantly, synthetic promoters acting like biological logic gates.
Logic gates, like A and D, or not in computing.
Exactly like that, but biochemical. The nanite doesn't just look for one marker. It might require the simultaneous presence of, say, 3 or 4 specific antigens to be absolutely sure it's found a cancer cell.
Ah, so multiple conditions must be met.
Right. And furthermore, the kill switch payload itself is often linked to that synthetic promoter, an engineered piece of DNA that acts like a digital F in command.
So if the nanite detects marker A and D, marker B and D, maybe a unique cancer-specific metabolic condition, then activate the kill switch.
Precisely. If those conditions are not met, the gene remains dormant, inactive. This multi-layered control, this biological two-factor authentication, if you like, ensures surgical precision.
That makes sense. That significantly reduces the risk of collateral damage to healthy tissues. Okay, so that's seek and destroy. But the architecture doesn't stop at destruction. The ultimate goal is reconstruction, right?
Absolutely.
Which brings us to the restore mode, or regeneration pathways. This sounds like the really ambitious part.
It is the great leap, no question. Restore mode uses the nanite not to kill, but to coax the body's own stem cells, or maybe specific progenitor cells, to replace damaged or lost tissue.
That's whoever way.
It delivers the code needed to reactivate deep-seated regeneration pathways, instructions that might have been entirely dormant, switched off since early childhood development.
Wow. Okay, that sounds fantastic on paper, but let's press on feasibility here. The source material talks about things like regrowing injured spinal cords. Right now, that kind of injury is catastrophic, permanent paralysis often. How could a nanite payload possibly achieve that level of systemic repair? It seems like a huge jump.
It's about delivering the complex genetic instruction manual for cellular renewal and reorganization. It's not just one gene. For spinal cord injuries, for example, the nanite wouldn't deliver a single fix-it gene.
No.
No, it would need to deliver the code for a whole cocktail of specific things: neurotrophic factors to encourage nerve growth, signals to guide the new axons, perhaps enzymes to gently break down scar tissue.
So it's coordinating a whole process.
Exactly. Its mission is multifaceted. Tell the glial cells surrounding the injury to stop forming that inhibitory scar tissue, which normally blocks regrowth, and simultaneously instruct nearby neural progenitor cells to differentiate, migrate, and bridge the gap.
So you're using the nanite to actively reverse the body's normal, often unhelpful reaction to severe injury.
And replace it with a full-scale guided rebuilding program. We're basically giving the body back the instruction manual it lost.
Could we see the same principles applied elsewhere? Like hearts, livers.
Absolutely. Mending scarred hearts after major heart attack. Right now, that scar tissue, fibrotic tissue, it doesn't contract. It weakens the heart.
Right.
Imagine nanites instructing progenitor cells to replace that non-functional scar tissue with healthy, contracting myocardium, or restoring livers fully after extensive cirrhosis damage, replacing fibrotic tissue with functional hepatocytes.
So we're essentially taking the body's innate repair mechanism, which normally just handles minor scrapes and bumps, and amplifying it, guiding it with a full genetic toolkit to tackle major systemic failure.
That's the goal. It could potentially stop the clock on many chronic degenerative diseases by restoring the original hardware, not just managing the decline.
Amazing. Okay, seek and destroy, restore. What's the third mode?
The third mode is arguably the most important for safety. It's the self-destruct mode.
Ah, the escape hatch.
Exactly. This ensures the nanite itself deactivates, degrades harmlessly, and leaves no trace once its therapeutic mission, whether that was destroying a tumor or restoring a damaged heart, is complete. It's the molecular equivalent of a clean shutdown and uninstall. No lingering code, no potential for long-term unforeseen effects.
That seems absolutely essential. Okay, this whole sophisticated design, the tri-mode architecture, seek and destroy, restore, self-destruct. It sounds like it's straight out of a lab 50 years in the future. How much of this is really grounded in current scientific context? Are these just nice ideas?
No, it's genuinely built upon existing tools that labs are using today. We have programmable CRISPR-based gene editors that allow us to cut and paste DNA with incredible, unprecedented precision. That's real.
Right. CRISPR is everywhere now.
We work daily with self-limiting viral vectors. These are delivery viruses that already have a predetermined lifespan built in. They're designed to stop replicating after a certain number of cell divisions or a certain time period. That exists.
Okay.
And as we just discussed, we have those synthetic promoters that act as biological logic gates, ensuring genes activate under very precise, specific conditions. Those are being designed and tested right now.
So the tri-mode architecture isn't inventing fundamentally new biology.
No, it's primarily the clever synthesis and integration of these existing modular tools. It's scaling them up, combining them into a highly controlled, repeatable therapeutic system. The components are largely on the shelf. The challenge is the system integration and ensuring safety at scale.
The synthesis, then, the big picture, is that the future medical system views almost every disease, from cancer to neurodegeneration to heart failure, not as this messy organic battle.
But fundamentally as a code delivery and execution problem.
Yeah. If we can write the right corrective patch, the right instructions, and ensure its safe, precise delivery and its controlled execution,
Then the body, using its own inherent mechanisms, corrects itself. We just provide the right information at the right time in the right place. And, that capability brings us logically, inevitably perhaps, to the next step, a vastly more profound step, maybe.
which is
Once you have a perfectly working, scalable, and importantly safe platform for DNA delivery and code correction, once you've figured out how to reliably fix all the bugs in the system, you don't just stop there, do you?
probably not. What comes next?
The next immediate, inevitable step is delivering upgrades, enhancements.
Ah, this is where we really leap from medicine, from fixing what's broken.
Into intentional directed evolution, augmentation.
If a viral nanite can deliver corrective programs to fix, say, cystic fibrosis,
Then it can certainly deliver constructive programs for traits that humanity, or at least that specific individual, never possessed before.
the sources use that kind of evocative analogy, don't they, about the predator species from the movies?
They do, incorporating the desirable traits of others to evolve beyond their current limitations. It's a powerful image.
But the analogy only goes so far,
The
right?
analogy holds in concept, but the methodology is crucial. It's fundamentally different. We are not performing crude organ transplants from other species or, splicing chaotic alien tissue into humans.
Yeah.
That's the old sci-fi trope.
Okay, so what are we doing?
We are identifying the specific genetic modules, the precise sequences of DNA responsible for a desirable trait in nature. Say, the extreme healing capacity of an axolot. Then we synthesize a version of that code from scratch, optimizing it specifically for human biology, making it compatible.
And then deliver it seamlessly via the nanite.
Exactly. It's more like borrowing nature's most effective solutions, translating them into human, rather than crudely stealing incompatible parts.
And the absolute genius of this approach, it seems, is that nature has already done billions of years of research and development for us for free.
Precisely. Evolution has solved countless problems we currently struggle with: survival in extreme environments, incredible sensory perception, rapid regeneration.
We just need to find the code, understand it, translate it carefully, and then apply it using these nanite delivery synthers.
That's the essence of it. Let's look at some of the specific examples the sources provide for borrowing nature's solutions, because they really map out a potential roadmap for human enhancement. It's not just vague ideas.
Okay, where do we start? Sensory upgrades.
Let's start there, yeah. Specifically enhanced night vision. This isn't just slightly better vision in the dark. No. The enhancement is achieved when the viral payload introduces genes encoding infrared-sensitive opsins.
Opsins. Those are the proteins in our retina that detect light, right?
Exactly. They're specialized photoreceptor proteins that convert photons of light into the electrical signals our brain interprets as vision. But standard human opsins only work within the visible spectrum, ROIGBV.
Red, orange, yellow.
Mm.
Yeah.
What this does is add new opsins sensitive to different wavelengths, specifically infrared. We're essentially expanding the operational spectrum of the human eye itself.
So the result isn't just better low-light vision.
It's vision extended beyond the normal visible spectrum. It would allow the recipient to perceive heat signatures directly, wavelengths that are currently completely invisible to us.
Like thermal goggles, but built into your eyes.
Essentially, yes. It could turn pitch darkness into a visible landscape of heat. Imagine the applications.
Yeah.
Or just the experience.
That is a staggering upgrade. Mind-blowing. But perhaps even more impactful on, say, immediate survivability or quality of life is the next one. Regenerative healing. We are talking about potentially installing something like a salamander-like limb regeneration pathway. I mean, that concept has been pure fantasy, pure science fiction forever.
It has. But the blueprint for immediate and complete self-repair exists in the animal kingdom. Axolotls, salamanders, they do it routinely. And crucially,
What?
elements of those regenerative pathways, the underlying genetic switches, still exist, dormant and unused, within our own human DNA. leftovers from evolution.
So the potential is there, just switched off.
Largely, yes. By delivering the precise genetic code via nanites, the right combination of transcription factors, growth hormones, signaling molecules, we could potentially reactivate those dormant developmental programs, the ones necessary for full regeneration.
So you're introducing the on switch and maybe the instruction manual that allows, say, a salamander to regrow an entire lost limb perfectly.
And instructing the human body's cells at the injury site to do the same. Replace lost bone, muscle, nerve tissue, blood vessels, skin, without the usual permanent scarring or the need for amputation. The nanites effectively press a molecular reset and rebuild button on damaged tissue.
The implications for trauma care, for amputees, it's almost too much to comprehend. Okay, what about surviving in difficult situations? Resource scarcity, extreme environments. The sources mention code inspired by diving mammals.
Right. This targets oxygen efficiency, optimization. Code inspired by animals like deep-diving seals or whales could potentially be used to optimize the structure or function of human hemoglobin.
Hemoglobin, the protein in red blood cells that carries oxygen.
Exactly. The goal would be to significantly increase the protein's ability to bind oxygen in the lungs, store it effectively, and then deliver it more efficiently to tissues throughout the body, especially under low oxygen conditions.
And the functional result would be?
A person could potentially sustain consciousness, and maybe even strenuous physical activity, with minimal error for significantly longer periods. Think about the massive advantage for deep-sea exploration or rescue, high-altitude mountaineering, surviving building collapses, or even just emergency medical scenarios where oxygen is limited.
Wow, okay. And finally, there's one more big one, essential for, say, the dream of becoming an interplanetary species. Radiation resistance.
Yes. This seems absolutely critical if we're serious about long-term goals like bases on the moon or voyages to Mars, or even dealing with potential nuclear accidents on Earth.
Where do we borrow that code from?
We look to nature's ultimate survivors, extremophiles like tardigrades.
Water bears, those microscopic things that can survive almost anything. Vacuum, boiling, radiation.
Exactly. They're famous for surviving levels of ionizing radiation that would instantly shred human DNA and kill our cells. They achieve this partly by producing unique DNA repair proteins. One famous example is called DSUP, which stands for damage suppression protein.
What does it do?
It seems to physically shield the DNA, almost like a molecular blanket, and also helps rapidly mend any breaks or damage that does occur from radiation.
So the idea is to take the gene for DSUP or similar proteins.
Synthesize a human-compatible version, integrate that genetic information into human cells via the nanite delivery system.
And we could drastically increase our cells' resistance to radiation damage, protecting our DNA.
That's the hypothesis. It could make long-term space travel, living outside Earth's protective magnetic field, fundamentally safer, reducing cancer risk, preventing cellular degradation.
Looking at all these examples, night vision, regeneration, oxygen efficiency, radiation resistance, here's the key unifying insight, isn't it?
What's that?
The nanite isn't creating these traits out of thin air. It's not magic. Each enhancement is simply biology repurposed, borrowed code.
Exactly. We are not inventing fundamentally new forces of nature here. We are learning how to identify, understand, borrow, and translate the incredibly clever solutions that evolution has already engineered over millions of years. We're applying those existing biological blueprints for human benefit. And that translation process, that careful adaptation, is what really differentiates this kind of intentional, directed program from the older, perhaps more chaotic ideas about genetic modification.
Right. We absolutely must clarify the distinction here. The source material calls it designing without, crossing the species barrier in a disruptive way.
It's a crucial point.
Because we remember maybe from early news reports the sort of clumsy days of genetic engineering, where scientists might say transplant a glowing gene directly from a jellyfish into a mouse, right?
The green fluorescent protein, yes.
Wow.
A useful marker, but a very direct transfer.
And that crude method, just taking a gene wholesale from species A and sticking it into species B, it risks all sorts of problems, doesn't it? Immune rejection, the gene not working properly, unstable expression, unpredictable side effects. The old way was fundamentally reactive and often, well, messy.
It could be. The synthetic method we're discussing here is profoundly different. We are not taking raw, foreign DNA from a tardigrade and injecting it into a human.
No.
No. Instead, we first understand the function of the desired trait at a molecular level, for example. How does that tardigrade DSUP protein actually protect DNA? What's the mechanism?
Okay, understand the how.
Then, armed with that understanding, We write an equivalent but fully optimized sequence of DNA from scratch, one that uses human-compatible codons.
Codons being the three-letter genetic words that our cellular machinery reads to build proteins.
Exactly. We design the gene so it speaks human, biochemically speaking. The analogy used in the sources is quite good. It's like translating a highly complex, nuanced poem into another language.
You don't just swap words one for one.
No. You'd lose all the meaning, the rhythm, the beauty. You have to preserve the core meaning and function, but you adapt the entire structure, the grammar, the vocabulary, specifically to the target language, in this case, human biology.
That is a critical distinction. It means you're aiming to get the benefit of the tardigrade gene without introducing something biologically foreign or potentially disruptive.
Precisely. It means that potentially superhuman traits like radiation resistance could theoretically be added seamlessly without triggering a massive immune rejection or turning people into unstable biological chimeras.
So the viral nanite in this model moves beyond being just a delivery truck.
It becomes more like a sophisticated scribe of tailored programs, delivering instructions written specifically for the recipient's operating system.
Okay. This more methodical synthetic approach, it lays the groundwork for what the sources call the modular design system. This is like the 5-step operational blueprint for how we might actually achieve intentional directed evolution.
Yes, it provides a framework for doing this systematically and hopefully safely.
Okay, step 1, building the foundation. That's the library of traits.
Right. This is envisioned as a massive, ongoing data collection and curation effort. It involves identifying and cataloging every potentially beneficial genetic module found across all of nature.
Everything useful.
Genes responsible for superior regeneration in amphibians, resistance to extreme heat or cold in microbes, enhanced senses in bats or birds, high-performance metabolic pathways in endurance animals. You name it.
So it's like creating the ultimate biological parts catalog.
Exactly. But indexed by functional module, what the gene does, rather than just by species. This library would need to be constantly updated, annotated, curated, forming the raw resource pool for potential enhancements.
Okay, you've got the library. Step 2 is making those exotic parts usable for us. Synthetic rewriting.
This is that translation process we just discussed. This is where bioengineers redesign those promising natural genetic modules for maximum safety, compatibility, and optimal expression, specifically within human biology.
Using techniques like codon optimization.
Yes, ensuring that when the new gene is read by human ribosomes, the resulting protein folds correctly, functions efficiently, integrates seamlessly with our existing biochemical networks, and, crucially, avoids triggering a strong immune rejection response. High efficacy, low risk.
Makes sense. Step 3 is then putting the newly written package together and ensuring it gets to the right address. Nanite delivery.
Exactly. This requires engineering the viral vectors themselves, the nanite shell, with extreme precision targeting capabilities.
So it's not just about injecting it generally into the bloodstream and hoping for the best.
Definitely not. It's about micromanaging precisely where that genetic payload lands. The nanite must be designed, perhaps using specific proteins on its surface, to only interact with and deliver its module to the specific cell types that require the upgrade.
Like only retinal cells for a vision enhancement.
Or only specific heart muscle cells for mending cardiac tissue. Or only certain types of neurons for a cognitive boost. It must deliver the code there and nowhere else. This pinpoint precision is vital to prevent chaotic, off-target effects or the code spreading where it shouldn't.
Okay, targeted delivery. Step 4 sounds absolutely critical, especially given the power we're talking about. Dynamic control.
This is arguably the cornerstone of safety and responsible use. It requires building robust regulatory mechanisms directly into the delivered DNA sequence itself.
Right.
We're not just dropping off code, we're dropping off code with built-in controls.
What kind of controls?
Things like molecular on-off switches. Perhaps the delivered gene is only expressed, only turned on, when triggered by an external, easily accessible, and harmless signal.
Like what?
Think about maybe a specific uncommon dietary compound, kind of molecular key that when you ingest it, it activates the expression of the new genetic program. Stop taking the compound, the effect fades.
Or maybe an external signal, like a specific frequency of light shown on the skin.
That's another possibility. This allows the user and their doctor to actively regulate the dosage, the timing, and the duration of the enhancement. It's not permanent unless you want it to be.
What else?
Furthermore, we'd need built-in time-limited expression features. Maybe the gene naturally degrades or switches off after a set period unless actively renewed. And most importantly, integrated safe rollback features.
A molecular undo button.
Essentially, yes. A way to quickly and reliably deactivate or even excise the introduced genetic module if there are unforeseen problems or the person simply changes their mind. Safety nets have to be integral to the design.
Absolutely. Okay, library, rewrite, deliver, control. What's the final step?
Yeah.
Step 5.
Step 5 recognizes that evolution, even directed evolution, isn't usually a single dramatic event. It's an ongoing process. So step 5 is iterative augmentation,
Meaning?
meaning we apply these upgrades gradually. We start small, perhaps. We monitor the effects meticulously in the individual, testing for safety, checking efficiency, seeing how well it integrates with their existing biology.
Learn as you go.
Exactly. Once an enhancement is proven safe and beneficial in that person, then maybe you consider stacking other enhancements over time, incrementally.
So the body, in this vision, becomes less of a fixed, static machine subject to inevitable decay?
And more like a platform for continuous updates, constantly improving its resilience, its capabilities, its longevity, like upgrading your computer's hardware and software over its lifespan.
The conclusion of this whole 5-step blueprint chapter, then, is pretty clear. This system, if realized, enables programmable evolution. It's intentional, it's directed by human intelligence, and it's incredibly fast compared to nature.
That's the key difference. Instead of waiting potentially millions of years for random mutation and natural selection to maybe yield a desired trait like radiation resistance,
Humanity could potentially test, optimize, and apply entirely new beneficial capabilities within a single generation, or even within an individual's lifetime.
we'd be applying the speed, the iteration, the design methodology of computing and engineering directly to the slow, organic process of biology. It's a paradigm shift.
Right.
But, and this is a huge but, whenever we discuss a technology that confers this much potential control over human biology, over life itself,
Yeah.
the gravity of the potential power is matched only by the severity of the risks involved. We absolutely must transition now to the mandatory safeguards and the immense challenges, technical and ethical, of deploying this kind of power responsibly. We can't ignore the potential downsides.
The technical dangers alone are, frankly, terrifying when you think about them properly.
They are.
Misprogramming just one of these nanites, getting the code slightly wrong,
Yeah.
you could inadvertently trigger the most immediate and fatal form of cancer, couldn't you? Inducing runaway growth in previously healthy cells.
That is a primary concern, yes. Off-target effects leading to malignancy.
And furthermore, what if a nanite fails its self-destruct sequence? If that uninstall program glitches?
That's another major risk. It could potentially propagate uncontrollably throughout the body, constantly expressing its payload long after it's needed, with unknown long-term consequences.
Or worse, could it potentially contaminate the environment if it somehow got out? Could we create a new engineered biological threat far more dangerous than any natural virus?
That containment and biosafety aspect is critical. And we also cannot forget the body's own defenses, the immune system. It's been honed over millennia to identify and attack viruses.
So it might just see the nanite, even a beneficial one, as an invader.
It might. It could simply overreact violently to the engineered viral vector itself, regardless of the payload, causing systemic catastrophic inflammation, a cytokine storm, even if the intended genetic program was perfectly benevolent and safe. The delivery system itself could be the point of failure.
Okay, those risks are substantial. What about the engineering solutions? How do we build in safety from the ground up? The source material insists on safeguards being integral, right?
Absolutely integral, not an afterthought, and it's adamant. Every single viral nanite must carry a compulsory, multi-layered kill switch, non-negotiable.
But isn't the challenge with the kill switch that the nanite is operating inside a human body? A complex, chaotic, constantly shifting biochemical environment? How do you engineer a universal stop signal that is guaranteed to work, that doesn't just get metabolized or accidentally blocked or missed?
You engineer redundancy and multiple failure points. You don't rely on just one mechanism. The kill switch, as envisioned, could be designed as a dual layer or even triple layer, fail-safe.
Like what? Give me an example.
Okay, first layer, maybe metabolic dependency. The nanite is genetically programmed so that it can only function, only replicate or express its gene in the presence of a specific rare nutrient or molecule that is not naturally found in the body.
So it needs an external supplement to work.
Exactly. The patient takes this supplement regularly to keep the therapy active. If they want to stop or something goes wrong, they simply stop taking the supplement. Cut the supply, the nanite shuts down and degrades naturally because it lacks a key component. That's one layer.
Okay, passive control, what's layer 2?
Layer 2, an active external trigger. Perhaps a specific harmless radiofrequency broadcast that penetrates tissue, or maybe a unique antidote molecule administered by a doctor if the nanite population starts acting unexpectedly or needs to be shut down immediately.
The doctor administers the antidote or flips the external RF switch.
And a secondary mechanism within the nanite triggers immediate self-destruction or inactivation. This level of belt and braces safety cannot be optional. It has to be designed in from the very beginning.
And presumably, the testing regime to prove that these kill switches work reliably has to be incredibly meticulous.
Absolutely rigorous. It has to be in far, far away from a living human being. The sources emphasize starting in organoids and lab-grown tissues.
Organoids, those miniature, simplified organs grown in a dish.
Exactly. We can build tiny, contained models of human organs, bits of lung tissue, functioning cardiac muscle, networks of brain synapses often derived from the patient's own cells, grown in a petri dish.
And use those to test the nanites first.
Precisely. We use these functional miniature biological systems to observe the nanite's effects in detail, check its dosage response, verify its targeting precision, measure its therapeutic effect, and, crucially, repeatedly and rigorously test the efficacy and reliability of the kill switch mechanisms under various conditions. It's essentially a biological simulation environment designed to fail safely outside the body.
Okay, so we manage the technical risks with kill switches and meticulous testing, but then we face the immense societal and ethical risks. The source material refers to this as the Great Divide. We face huge ethical questions regarding access, don't we? Who gets this technology? Will it be affordable? And what about abuse? And the terrifying potential to fracture society, maybe permanently, into the enhanced and the left behind.
It's a profound concern.
I mean, if true radiation resistance, or full limb regeneration, or significantly extended lifespans become a reality, but they're a luxury available only to the extremely wealthy or the powerful, what does that do to global social cohesion, to our sense of shared humanity?
That is probably the defining ethical challenge of this entire technological trajectory. And the source material, to be clear, doesn't pretend to offer easy, simple answers, but it absolutely mandates proactive solutions. We need to be thinking about this now. Such as the urgent need for global ethical frameworks to be debated and established now, well before the technology reaches full maturity and widespread deployment. This requires international cooperation, difficult conversations.
What should those frameworks cover?
Things like global consensus on equitable access and distribution models, mandated open source access, perhaps to core therapeutic research findings, preventing essential cures from being locked behind exorbitant paywalls. And crucially, clear, enforceable international treaties defining and strictly limiting non-therapeutic military or coercive applications. We need rules of the road, agreed upon globally.
The stated goal of such frameworks must be
to ensure the technology primarily uplifts all of humanity by curing devastating diseases, ending genetic suffering, maybe improving resilience for everyone rather than becoming yet another powerful engine of social stratification and economic division. It's a tall order.
It really is. Yet, despite all these staggering hurdles, the technical dangers, the societal divisions, the ethical minefields, the ultimate conclusion, according to the sources we reviewed, seems, well, almost unavoidable.
It feels like an established trajectory, yes. The foundational principles are falling into place. DNA is information, a programmable code. Viruses are near-perfect biological couriers for that information. And cells are fundamentally programmable biochemical machines.
The convergence is powerful.
The convergence of these facts means humanity is rapidly approaching the capability, whether we're fully ready for it or not, to literally rewrite its own biological operating system.
And the first targets will be the obvious ones?
Diseases like cancer, genetic disorders, they will likely be the first casualties, the first major bugs to be debugged, like corrupted files being repaired or deleted. Aging itself will likely follow, perhaps significantly delayed, maybe even partially reversed, by regenerative modules delivered systemically throughout the body.
But beyond just fixing what's broken, beyond repair and maintenance, lies the ability to design, to create.
Yes, the deliberate crafting of new, chosen capabilities, the upgrades we detailed earlier, the enhanced senses, the regeneration, the resistances.
So the human body in this future potentially moves from being seen as this fixed, fragile, inherited vessel
to becoming a platform. A biological platform capable of receiving continuous updates. Customizable, adaptable, resilient, and perhaps enduring in ways we currently only dream of.
We are moving, it seems, from merely attempting to fix our biology when it breaks
to actively, intentionally co-authoring it, deciding what it means to be human in the next stage of our evolution. Hashtag outro.
Yeah.
So, to synthesize the journey we've taken you on today, humanity seems poised, technologically at least, to shift from merely surviving what nature and chance deal us biologically
Yeah.
to becoming the active co-authors of our own biological destiny. It's a shift in agency.
Diseases, in this view, will eventually be rewritten, debugged, not just endured or managed.
Disabilities could potentially be patched, corrected at the source code level, not just accommodated.
And new abilities, enhancements might eventually be chosen, designed, installed, not just left to the incredibly slow, random lottery of natural mutation over millennia.
Viral nanites, these reprogrammed viruses carrying precisely coded DNA instructions, they are the potential biological quills, the molecular pens with which humanity might write its next evolutionary chapter.
It really represents the moment where healing and evolution start to merge, doesn't it? Where the perceived biological limits of the flesh might begin to give way to the designed ingenuity of the mind.
It does. And the virus itself, for so long the ultimate symbol of death and decay, is transforming, conceptually at least, before our very eyes, into a potential scribe of life, maybe even extended or enhanced life.
Which leads us then to our final provocative thought. Something for you, the listener, to consider as you process this deep dive we've taken.
If we can truly master this programmable evolution, if we reach the point where we can reliably install salamander-like regeneration, if we can upgrade our vision to perceive entirely new spectra of reality, if we can integrate resistance to the worst hazards of deep space,
Then what began as a mission to find a cure?
will rapidly, perhaps inevitably, end as an act of creation, of self-creation. So the question for you is this. Are we, through this technology, merely unlocking the vast, untapped potential already encoded within the immense library of life on Earth? Or are we, by taking the reins of our own biology, fundamentally and perhaps permanently, changing the very definition of what it means to be human?